What's hanging, YouTube? Today, I'll be doing a rhetoric presentation about cloning. Now, you may be asking, what's this context that you have from commentary and video games and whatever you do that's probably dumb on your channel? Well, I'm going to tell you one thing. This presentation is especially hard to me because I'm going to be impersonating a YouTuber, and you know that my impersonation of YouTubers are 100% accurate to the most finite details.
Did I lie?
Now, today's impersonation, or person I'm going to be trying to impersonate, is Physics Girl, who basically has a YouTube channel here. Now, today's science topic, since Physics Girl does mostly science topic, I hope, is about cloning.
Should I make these videos or should I not? That is the true question.
Now, I use the source National Humane, I read that wrong.
Buttermilk biscuit.
National Human Genome Research Institute, and they give a definition of cloning, and they basically have a Q&A of basically whatever questions you had about cloning. Now, in nature, apparently cloning happens. I mean, that's where you get identical twins. Miley Cyrus and Noah Cyrus. Wait, those aren't identical twins.
Buttermilk biscuit.
Although, I kind of see similarities between them, oddly enough. FBI, open up! And there are artificial cloning. Now, apparently in this website, there are 3 artificial types of cloning. I said that wrong. I meant types of artificial cloning.
Buttermilk biscuit.
There is the reproductive cloning, there is the therapeutic cloning, and there is the gene cloning. Now, at first I thought the therapeutic cloning was a little funny because I was like, what's so therapeutic about being cloned? And then, but when I read down a little further, I obviously saw how therapeutic cloning is, which is almost ironic in a sense. Now, your boy obviously researched this very well, so make sure you can follow along a little bit. Therapeutic cloning basically is when you take a damaged tissue or perhaps a diseased tissue and clone it so that it hopefully can cure that disease or damaged tissue. Gene cloning basically produces clones of a specific segment of DNA. And I believe the last one was, what was it? Reproductive cloning, and that basically means cloning a whole animal. I know, surprising, right? Now, how are genes cloned? Now, apparently in the National Human Genome Research Institute, they say, researchers routinely use cloning techniques to make copies of genes that they wish to study. The procedure consists of inserting a gene from one organism, often referred to as foreign DNA, into the genetic material of a carrier called a vector. Oh, are we talking about the vector from Despicable Me?
Oh, poop.
No, I doubt it. Examples of vectors include bacteria, yeast cells, viruses, or plasmids, which are small DNA circles carried by bacteria. Basically, hockey in 30 seconds. After the gene is inserted, the vector is placed in laboratory conditions, which is pretty much pristine cleanness, where you can't literally drop any single liquid, that prompt it to multiply, resulting in the gene being copied many times over. So basically what they're saying in a nutshell is that they just create a situation inside of a gene itself, and then basically have the clone multiply on itself so that they get the desired amount or the desired product that they have at the end. Now, the next question that the National Human Genome Research Institute answers is, how are animals cloned? And apparently they remove from the test animal that they're going to clone what's called a mature somatic cell, which is can be a skin cell. And it basically says that any cell within the body that provides significant feature, they will take that out so that they can clone it in another dimension. Not another dimension, but in a laboratory sense. Now, researchers can add some DNA to the somatic cell, to the empty egg, in 2 different ways. The first method, they remove the DNA containing the nucleus, of the somatic cell with a needle and inject it into the empty egg. So basically catch and release. So they catch the nucleus and then they shove it into the egg. And then the second way is that they use an electrical current. Quite ironic that they use electrics in biology, but hey, I'm not the one to judge. In order to fuse the entire somatic cell with the empty egg. So basically, fusion.
Fusion. Ha!
Yes, you're doing it! Stay with it!
You don't know the way.
Yes, I
Do you know the way?
Basically, though, both end up having an early stage embryo in the test tube, and then that is implanted in a womb, and then you pretty much get the process from there. Now, the next question that the National Human Genome Research Institute obviously answered for me is, what animals have been cloned? Oh boy. It says that basically over the last 50 years they try to clone stuff, but it didn't work out because often the test subjects would die within a quick amount, and that often the animals are not suitable enough in order to live because they have some genetics that are missing, or just simply small factors in the biology of that genome that was implanted in the embryo missing, and that often leads to a catastrophic mutation and most likely death. It wasn't until 1996 that researchers, though, succeeded in cloning the first mammal from a mature somatic cell, as I explained earlier, taken from an adult mammal or animal. After 276 attempts, boy, did this get me some Guardians of the Galaxy vibes from Volume 2. After 276 attempts, Scottish researchers, yes, the Scottish people, the Scottish researchers finally produced Dolly. That's right, Dolly the sheep, or as in that time, Dolly the lamb, from udder cell of a 6-year-old sheep. Basically, what that means is they took the somatic cell from that 6-year-old sheep in order to produce Dolly the sheep. 2 years later, researchers in Japan cloned 8 calves from a single cow, but only 4 survived. So pretty much it's like a 1/2 survival rate here. Besides cattle and sheep, other mammals have been cloned from somatic cells include cat, deer, dog, horse, mule, ox, rabbit, and rat. In addition, a rhesus monkey has been cloned by embryo splitting. Now, we come back to what the main question is, and the question that we've all been asking for many centuries. Not many centuries, because we didn't have cloning for a while. But we've all been having the question, can a human be cloned? And it seems so far that our biology does not provide that. Human cloning, in the words of the scientists at the National Human Genome Research Institute, say that cloning a human is not possible, unfortunately. And mainly that is because the human often has very relatable features that are very complex, and often taking a simple somatic cell from a mammal or just a simple human itself will produce often mutations that will not often be suited as humans, or perhaps even lower. But tests have been run, and often these test subjects haven't been surviving for long. I'm saying this almost as if like I'm an evil doctor from Bowser, or perhaps Sonic Eggman, right? Now, the other types of cloning goes over simply just what type of therapeutic cloning there is, which is obviously used for medical purposes in order to heal wounds, which is actually very nice. And yeah, that's pretty much the gist of cloning within the National Human Genome Research Institute. Now, my personal opinion about cloning is very simple. If our biology can advance it so much that we can clone a human, there will obviously be concerns about what will happen with the clones, or perhaps what rights the clones will provide, since they literally just came from the test subject itself. and that will be a lot more complicated than expected. But hopefully, if we have that technology, we will hopefully resolve that type of philosophical question soon. This has been an episode of Physics Girl impersonation that Kevin has done terribly. Thank you for watching, and I'll see you in probably the next video, which will hopefully be when I respond to Ella Harper. So yeah, peace out.